Prokaryotic cells, bacterium. Prokaryotes, kingdom Monera, so called monerans are prokaryotes. और इसमें includes है बैक्टीरिया, साइनोबैक्टीरिया, ब्लू ग्रीन एल्गी, माइकोप्लाज्मा, पिपीलो, प्लीरोनिमोनिया, लैक ऑर्गेनिस, स्पाइरोकेटिस and rickettsia. Okay, let's study bacteria. Bacteria. Among the prokaryotes, bacteria are simplest and commonest type of organisms, found in almost all the places, in the hot springs, beneath of icebergs, in intestine of man, deep in the soil, deep in soil water, etc. Or first discovered by Anton von Leeuwenhoek in 1675. Bacteria. Okay. Size of bacteria vary greatly in their size. A smallest bacterium is dialyster 0.15 to 0.3 micrometer in diameter, and largest bacterium is a spiroquete about 500 micrometer. Normally size of bacillus bacteria lies from 0.3 micrometer to 15 micrometer. याद रखेंगे एग्जाम के लिए डायलिस्टर स्मॉलेस्ट बैक्टीरियम, लार्जेस्ट बैक्टीरियम स्पाइरोकैट. ओके. Four basic shapes of bacteria. Ejecon recognized four basic shapes of bacteria. Coccus, spherical shaped bacteria. Bacillus, rod shaped bacteria. Spirillum, spirally coiled and flagellated bacteria. Vibrio, comma shaped and flagellated bacteria. And coccus जो बैक्टीरिया है वो देखेंगे. सिंगल राउंडेड बैक्टीरिया को कहते हैं माइक्रोकोकस. ओके. टू राउंडेड बैक्टीरिया डिप्लोकोकस. फोर राउंडेड बैक्टीरिया टेट्राकोकस. Chain of cocci, streptococcus, and bunch of cocci, staphylococcus, and 8-celled cubical mass of bacteria is called as sarcina. And bacillus, diplobacillus, palisade bacillus, streptobacillus. Okay. Examples of bacteria on the basis of their shape. Monococcus, roseus. Diplococcus, pneumoniae. Tetracoccus, Neisseria gonorrhoeae. Streptococcus lactis. Streptococcus pyrogens. Staphylococcus aureus, sarcina. Staphylococcus lutea. Monobacillus, anthracis, and Lactobacillus lactis palisade. Bacillus, Corynebacterium diphtheriae. Streptobacillus, Bacillus cereus, Spirillum volitans, and Treponema pallidum. Vibrio cholerae. Okay. And next we see that on the basis of cell wall, bacteria are divided into two types. Number one is the gram positive and number two is gram negative. Okay. And the gram positive bacteria. Procedure of gram staining. Bacteria stain for 30 minutes with purple colored crystal violet called gram stain. Okay. Washed with water and treated with iodine acts as mordant. And washed with alcohol decolorizing agent and stained with pink colored saffron dye, a counter stain. Then we see that some bacteria purple colored gram positive and some bacteria pink colored, that is gram negative. Okay. So gram positive bacteria is purple colored and gram negative bacteria is pink colored. Differences between gram positive and gram negative bacteria. Staining ability: absorb gram stain and appear purple colored. Gram positive don't absorb gram stain. Gram negative. Monolayered, mainly of murine, gram positive, and two-layered, outer of glycolipids and inner of murine, gram negative. Thickness of cell wall, gram positive 100 to 200 एंगस्ट्रॉम, ग्राम नेगेटिव का सेल वॉल 70 to 120 एंगस्ट्रॉम थिनर होता है. म्यूरिन परसेंटेज ग्राम पॉजिटिव में 70 to 80%, जबकि ग्राम नेगेटिव में म्यूरिन जो है वो मात्र 10 से 20% होते हैं. लिपिड्स जो होते हैं ग्राम पॉजिटिव में लेस होते हैं, ग्राम नेगेटिव में ज़्यादा होते हैं. मेसोजोम्स ग्राम पॉजिटिव में प्रेजेंट होते हैं, जबकि मेसोजोम्स ग्राम नेगेटिव में एब्सेंट होते हैं. पीली जो है वो ग्राम पॉजिटिव में एब्सेंट होते हैं और ग्राम नेगेटिव में प्रेजेंट होते हैं. एग्जांपल्स हैं ग्राम पॉजिटिव के डिप्लोकोकस निमोनियाई, और ग्राम नेगेटिव का एक एग्जांपल है इसेरिया कोलाई. याद रखेंगे, इसेरिया कोलाई ग्राम नेगेटिव बैक्टीरिया एंड डिप्लोकोकस निमोनियाई इज ग्राम पॉजिटिव बैक्टीरिया. बेसिक स्ट्रक्चर ऑफ ए बैक्टीरियल सेल. सेल इन्वेलप. It is outermost covering of the cell. It provides shape and rigidity to the cell. It also protects from mechanical injuries and bursting or collapsing. It is formed of three basic layers, each of which is specialized to perform a specific functions: glycocalyx, cell wall, and plasma membrane. Okay, A. Glycocalyx. Outermost layer and varies in chemical composition and thickness in different bacteria. In most of bacteria, that is Escherichia coli, it is in the form of a loose mucilaginous covering and is called a slimy layer, which protects the bacterium from chemical dissociation, action of phagocytes, and help in adhesion. But in some bacteria, it becomes hard and tough and is called capsule, okay, which resists phagocytosis. Capsule, okay. B. Cell wall. It lies inner to glycocalyx. Lies inner to glycocalyx. Cell wall. It is rigid, protective, and supportive. In Escherichia coli and gram-negative bacteria, cell wall is two-layered. A. Inner layer of murine or peptidoglycan, and B. Outer layer is made up of glycolipids. In the gram-positive bacteria, cell wall is a single-layered. Okay, gram-positive में cell wall single-layered and is made up of only murine, mainly murine. Plasma membrane. It is innermost layer of cell envelope. It is thin, transparent, and semi-permeable membrane. It is lipoproteinous and trilaminar, three-layered like that of eukaryotes. It is also called respiratory membrane, as in which with components of electron transport chain and enzymes of respiration. It regulates the exchange of specific materials between the cytoplasm and extracellular medium. It also has certain receptor molecules which help the bacteria to detect and respond to chemicals. Mesosomes and chromatophores. Look, mesosomes and look, chromatophores. Mesosomes. These are characteristic features of prokaryotes. Characteristic features of prokaryotes is mesosomes. Okay, formed by infolding of plasma membrane in only gram-positive bacteria and may be in the form of vesicles or tubules or lamellae. These help in following functions of bacteria. These help in replication of DNA and their separation into daughter cells during binary fission. These increase the surface area of respiratory membrane. These help in cellular secretion and these help in cell wall formation. Chromatophores. These are internal membrane system, help in increasing the surface area for increased enzymatic activity and metabolic rate. In cyanobacterial cells, these contain pigments. Cyanobacterial cells में ये contain करते हैं pigments. Okay. Cytoplasm. Now let's study the cytoplasm in bacteria. Cytoplasm, it is colorless, translucent, and viscous ground substance. It present inside the cell envelope. It has reserved food in the form of glycogen and lipids, and it has 70 S types ribosomes. Okay, membrane-bound cells organelles like mitochondria, endoplasmic reticulum, Golgi apparatus, plastids, lysosome, etc. are all together absent from the cytoplasm. और यहाँ मेम्ब्रेन बॉन्ड ऑर्गेनल्स नहीं पाए जाते हैं, माइटोकॉन्ड्रिया नहीं पाए जाते, एंडोप्लाज्मिक रेटिकुलम, गोल्गी apparatus नहीं पाए जाते, plastids and lysosomes are not found in the cytoplasm of bacteria या prokaryotes. The ribosomes may be in the free form of or arranged in the form of helical series on single messenger RNA strand, especially during protein synthesis, called polyribosomes or polysomes. Note down polyribosomes or polysomes during protein synthesis. Okay, each 70S ribosomes is 15 nanometer by 20 nanometer in size, and each 70S ribosome is formed of two subunits, 50S and 30S subunits. And cytoplasm of bacteria also has some non-living structures called inclusion bodies, that is reserved food, phosphate granules, etc. While inclusion bodies of cyanobacteria also include cyanophacian granules, glycogen granules, and gas vacuoles. Nucleoid. Nucleoid. It is incipient nucleus. Nucleoid is incipient nucleus of the prokaryotes. As a well organized membrane bound true nucleus is absent in them. और यहाँ well organized membrane bound true nucleus absent होता है. It has single and circular chromosomes with no free ends, which is formed of double-stranded DNA but is not associated with histone proteins. It is also called genophore or prokaryon या प्रोक्रोमोजोम. नोट डाउन न्यूक्लियाड को हम लोग also कहते हैं जीनोफोर, prokaryon या prochromosome. This circular DNA is extensively looped and coiled with the help of nucleoid proteins. It lacks nucleoplasm. Nucleoid lacks nucleoplasm, nucleolus, and nuclear membrane. In some bacteria, that is Escherichia coli, additional double-stranded circular DNA molecules called plasmids are also present. Each plasmid contains a few thousand genes, so act as extra nuclear genetic material. The plasmid DNA provides certain peculiar phenotypic characteristics to these bacteria, that is fertility or sexual factor, antibiotic resistance, etc. Fertility factor controls the formation of pili. Okay, flagella. Bacterial cells may bear one or more long, thin, and whip-like protoplasmic structures called flagella. Each flagellum is monofibrillar filament of globular protein flagellarin molecules. It is differentiated into three parts. Number one, basal body or shaft. Number two, hook. And number three, filament. Flagella. Basal body lies within the cell wall and is formed of two M and two S rings in the flagellum of gram-negative bacteria, while it is formed of one M and one S ring in the flagellum of gram-positive bacteria. Depending upon the number of flagella, the bacterium are following types: monotrichous, amphitrichous, or cephalotrichous, and lophotrichous, both sides, peritrichous on full body, and atrichous, no flagella. Okay, atrichous, flagellum absent, that is cocci bacteria. Monotrichous, single flagellum at one end, that is lactobacillus lactis. Amphitrichous, amphitrichous, two flagella, one at each end, that is Spirillum serpens, and cephalotrichous, two or more flagella at one end, that is Pseudomonas fluorescens. Okay, and next is Lophotrichous, two group of flagella, one at each end, that is Baronella bacilliformes, and Peritrichous, several flagella distributed on whole of bacterium, that is Escherichia coli. Okay, pili or fimbriae. Now let's see that these are the pili and these are the fimbriae. Okay, in certain bacterium, that is Escherichia coli, minute hair-like small site and narrow structures called fimbriae, or long tubular structures called pili. Sex pili, as the pili of male bacterium formed of pilin protein, fimbriae, are not involved in motility but help in adhesion of bacteria to the rocky substratum or to the host tissue. Now, differences between fimbriae and pili. Fimbriae or pili में differences both in gram-negative and gram-positive bacteria. Fimbriae and pili only in gram-negative bacteria. Fimbriae more 300 to 400 per cells, pili less 1 to 4 per cells. Fimbriae shorter and narrow, where pili longer and broader. Fimbriae function in adhesion and pili function in conjugation. Okay. Now, eukaryotic cells we study in the next class. Thank you.